Yes, so this is now for something completely different. If you thought the last talk was different, this will be, I think, very completely different than what you've heard already today. So I just moved to the University of Pittsburgh a little over a year ago from Washington University, and I'm the director of molecular imaging at the Department of Radiology, and so I'm going to talk a little bit about one of the projects that we're working on. And most of this work was actually done at Washington University. Our preclinical imaging facility at Hillman Cancer Center just opened yesterday. So we'll be doing lots of exciting stuff here as well. So I'm going to start just kind of from the beginning about molecular imaging. And it's not really been framed so much in more of the practical sense. I think many of the talks, if not all, have a component of molecular imaging. But in terms of how do we apply this to imaging of disease, and this definition was made by the Society of Nuclear Medicine and Molecular Imaging about 5 years ago now. But molecular imaging is the visualization, characterization, and measurement of biological processes at the molecular and cellular levels in humans and other living systems. So I think what's really important to note is that it's not, and I mean, again, today we've really been delving into the math. I'm going to not really delve into the math at all, but just to let you know that in the practical sense, when we're doing imaging of humans, we're looking at pretty pictures, of course, but we're really trying to characterize disease, not to just look at the anatomy, but to actually tell us something biochemically and physiologically about what's going on. And then I think the most important is that we're trying to quantify this. So we want to look at disease and we want to be able to image and quantify disease, maybe before treatment and after treatment would be one example. So molecular imaging, it can be used in early diagnosis and identification of functional changes in tissues. We can look at individual patient management in real time, which is personalized medicine that we've all heard so much about, and it can also facilitate drug development. So molecular imaging, I should say maybe more what it doesn't include, which is some of the stuff that's been talked about here, is CT, just traditional anatomical imaging is really not considered to be molecular imaging. Standard magnetic resonance imaging of anatomy is really not molecular imaging. So what we're really looking at is positron emission tomography or SPECT, single photon emission computed tomography. This is the traditional nuclear medicine imaging. Ultrasound, we heard about some stuff that Kang Kim was talking about with maybe photoacoustic imaging as well with the contrast agent. Fluorescence, optical, and bioluminescence imaging. But nowadays it's really the fusion imaging, and we've heard a lot of talks today about the multimodality and really how it's important to combine modalities. And the anatomical imaging is really critical in doing this because I think it helps us to more accurately quantify what we're seeing. And so now we have PET-CT, SPECT-CT, and more recently PET-MRI. And we just had a PET-MRI has been installed, or MR-PET, depending on what side of the equation you come on. I'm on the PET side. That was installed back in May, and I think they're going to get started on their first patients very soon. Magnetic resonance spectroscopy is also another molecular imaging modality. So I'm going to really be focusing on PET and with CT and really more on the preclinical, so in small animals. And since nobody's mentioned PET today that I've seen, I have missed a few talks, but essentially the way PET works is PET requires radiopharmaceuticals. So you have to have a molecule of some sort that's radio-labeled with a radioactive, a radionuclide that emits positrons. And what a positron is just a positively charged electron. So if you have your positron-emitting isotope, this collides with a negatively charged electron, and that this annihilation radiation results in two 511 keV gamma rays that are approximately, but not exactly, 180 degrees apart. And so the PET scanners, which were developed back in the 70s, what they see then are they see other things, but they mostly see the coincident 511 keV gammas. And that's why this is, because you're just really concentrating on the coincident emissions. That's why PET is so much more quantitative than traditional single photon emission computed tomography or gamma scintigraphy. So now pretty much a standard now for most PET imaging is that it's combined with CT. This provides very high accurate co-registration, and it also enhances the resolution of PET and makes it fully quantitative. So you can see here that here's just the FDG. An FDG is a fluorine-18 labeled deoxyglucose molecule, which is taken up in highly metabolic tissues such as tumors. And so if we have the FDG PET scan here, we can co-register this with a CT scan, and we can really then focus on where the hotspots are, quantify them, and look at these over time, maybe monitoring treatment. So I'm going to really be focusing on molecular imaging probes, agents, radiopharmaceuticals today, because that's what I actually have a chemistry background, and that's what my lab focuses on. So the molecular imaging agents, again, they can be radiopharmaceuticals, MR contrast agents, optical imaging agents, gold nanorods, such as what Kang Kim talked about. These are used to help to visualize, characterize, and measure the biological processes in living systems. And so these can be endogenous molecules, such as used in magnetic resonance spectroscopy, but most of the time when we talk about molecular imaging agents, we're really looking at things that are injectable or inhaled, such as the fluorine-18 labeled deoxyglucose for PET. Here's an optical dye conjugate that can be used for optical imaging. And then again, these are the endogenous molecules that will change over disease state in magnetic resonance spectroscopy. So what we do in my lab is we kind of look at all phases of the molecular imaging agent design cycle. So everything that we do in my lab starts with chemistry. We have to synthesize targeting molecules that will target different disease states. And typically, these are going to be agents that we can radiolabel with radionuclides, but we also do put dyes on them for either validation with histology or some optical imaging. We first then evaluate our agents in in vitro systems. That could be some sort of a biological assay or cells grown in cell culture. If this pans out and we find out that our compound truly does bind to the receptor that we're interested in, we then move to preclinical imaging in vivo. And this is, in my lab, this is typically in rodent models, although we do have a project that's ongoing at the University of Pittsburgh with Joanne Flynn that involves imaging of granulomas that are the hallmarks of tuberculosis in a non-human primate model. We always have to validate what it is we're seeing. We might see hotspots. They may correspond with what we want to look at that we're trying to image, but we don't really know that we've hit the target. So we always, when we're designing and developing an imaging agent, we must make sure that we histologically validate what it is we're trying to image. If all of this works, then we're ready to go into humans. And these little words, IND submission, this can be it's hundreds of thousands of dollars. It could take a couple of years if everything goes really well. So then we get into humans and we do our clinical imaging, and then we find out that maybe our compound really isn't so good. It looked great in the mouse and maybe even in a larger animal, but we get to humans, it really didn't do what we wanted, and so then we start all over again. We make new compounds and we go through the cycle again. So my lab, I said I was a chemist. I'm an inorganic chemist, so I started my career in nuclear medicine by focusing on metal radionuclides, which is probably why our labs are next to the hot metal bridge and down on the river down south. So, but we work a lot with copper-64. This has a fluorine-18 that you may have heard more about with FDG imaging, has a 2-hour half-life. You can do quite a bit with a 2-hour half-life, but you can do a lot more chemistry with a 12.7-hour half-life. So we can actually get this isotope shipped in, and we can get a lot of work done with this. And so what's great about copper-64 is that it emits positrons for the PET imaging. It also emits beta minus, which are negatively charged electrons, which are very good for therapy. When I worked at Washington University, I was spoiled because we were able to make it right there on site. And we still do purchase it now from Washington University, but we also get it from the University of Wisconsin. And so what we do with this copper 64, one focus area in my lab is to look at how we can take this radiometal, put it into a chelator, and make sure that the metal chelate is very stable in vivo. And then we attach this metal chelate to some sort of biologically targeting molecule. This could be a peptide, peptoid, peptidomimetic, small molecule, antibody, nanoparticle. We've looked at all of them. And so, but the key thing about this biologically targeted molecule is it must bind to some sort of protein that's expressed usually on the surface of a cell that may be upregulated in some disease state. And my lab really mostly focuses on cancer, although these receptors are also overexpressed in obviously in neurodegenerative diseases or in cardiovascular disease and even in TB, which we're working on now. So as I mentioned, we focus on developing these chelators. We also want to make sure when we develop these radiopharmaceuticals, we want to be able to get this radiometal inside this compound very quickly because we do have decay going on and we have a limited amount of time. We want to make sure it's stable. If the minute we inject the compound into the body, it immediately falls apart, we've accomplished nothing. And that's actually fairly tricky with copper. It's not so tricky with other metals like even gadolinium, or although the MR people know that there have been issues with stability of gadolinium chelates, which is very toxic. But there are many metals that are more easy to work with. Copper is a little trickier. We want to make sure that whatever we've done to this targeting molecule binds to this receptor in vivo and is not hindered by that. And we also want to make sure that this compound doesn't stick into the liver or the kidneys, the rest of it just kind of usually is excreted very quickly. So given that background, what I'm going to focus on today is what we're trying to do in targeting imaging metastasis, or actually imaging the metastatic process and maybe even what happens before cancer metastasizes. So I don't think I have to, I think pretty much everybody in the room knows, has had a relative, a good friend die of cancer, and typically it's not the primary tumor that causes the mortality. A lot of times, although there certainly are cancers where that is, people do die of that, but typically the primary tumor is such that you can remove it and surgically remove it, and oftentimes it won't go back. But what happens, especially in things like breast cancer and colon cancer, even prostate cancer, is you remove the tumor and then you still get metastatic disease, and this is really what people die of. And in the case of breast cancer, only 20% of people who have metastatic disease live 5 years or longer. Colorectal cancer, if you have metastatic disease, only 10% will live 5 years or longer. So clearly this is a very important problem that it would be great to be able to find out in advance. You're doing surgery on the primary tumor. Who is it that might metastasize? You can find out things about the tumor maybe histologically, but there's a lot of other things going on in the body. And could we really pinpoint who those patients are before this metastatic process happens? And so a long time ago, well over 100 years ago now, Stephen Paget came up with the seed and soil hypothesis. And we, even maybe many of us who, as far as we know, don't have cancer, we may still have circulating tumor cells floating around in our bone marrow, in our blood. Hopefully not, but, and especially if you've had cancer, they check now for circulating tumor cells or disseminated tumor cells that are in the bone marrow, because that kind of makes it, that maybe make you more likely to have a metastasis. And so well over 100 years ago, before anybody really understood this process, Stephen Paget he noticed that circulating tumor cells only seed where there's congenial soil. And so what they now call this is the tumor microenvironment. And so you have to have a really receptive environment for these metastatic tumor cells to go to a distant site and actually form metastatic tumor deposits. And so what we're interested in is can we image the soil? Can we find out whether or not the environment is hospitable to tumor cells? And can we find this out before the metastasis actually happens? So about, I guess it was, the first paper came out in Nature in 2005 by David Leiden's group at Cornell Medical College, where he coined, I don't know if it was him or if this term had been around before, but he really showed that there seems to be a pre-metastatic niche. And what he showed, and this is from his paper, is that what you have is you have bone marrow-derived cells, and these express different receptors such as vascular endothelial growth factor and this very late antigen 4. So they have these bone marrow-derived cells like hematopoietic progenitor cells. And he showed ex vivo, not in vivo, but he showed ex vivo that using histological methods and flow cytometry, that these cells home to sites of metastasis in a mouse model before tumor cells actually arrive. And so first you have the pre-metastatic niche that forms clusters of bone marrow-derived cells. Then the tumor cells then start arriving at this site, and they form micrometastases. And then after that, that's when you get your angiogenic elements. The blood vessels start to form. You have endothelial progenitor cells that come in, and that's what you need for the completion of this metastatic lesion. And so what we really wanted to focus on is, is it possible to image these clusters of the bone marrow-derived cells before the tumor cells actually arrive? Or can we see if this is possible? And we wanted to do this with kind of a combination of PET and optical methods. So we decided to focus on the very late antigen 4, which is also called alpha-4 beta-1 integrin. This is it's an integrin. It's a protein that's upregulated in tumors and bone marrow-derived cells. So integrins are actually it's a family of cell adhesion molecules, and there's over 20 different combinations of the alpha and the beta subunits. They're heterodimeric proteins. They play this alpha-4 beta-1 specifically plays an important role in lymphomas, multiple myeloma, melanoma, and cancer metastasis and development. It's a key player in drug resistance of both multiple myeloma and acute myelogenous leukemia. And it is putatively upregulated in the hematopoietic progenitor cells involved in the formation of the pre-metastatic niche. So our study objective was to see if we could address an imaging strategy for in vivo imaging of these hematopoietic progenitor cells at pre-metastatic sites before a secondary tumor is established. And this work was primarily done at Washington University with my co-investigators shown down here. And we addressed the hypothesis that the very late antigen 4 is a biomarker for hematopoietic progenitor cells that might predict future sites of metastasis. So we, in the literature, that's why we decided to focus on targeting very late antigen 4. We also have plans in the future to look at the VEGF R1 receptor ligand. There right now isn't a very good ligand, but we are planning to do ligand discovery to come up with a specific ligand for that. So we initially focused on this VLA4 target, and there is a very high affinity ligand that was already developed by Kit Lamb's group at UC Davis. And we initially collaborated with the Lamb group, and we put the cross-bridge chelator on there for radiolabeling with copper-64. And we did a preliminary study with them, and that was published a few years ago, showing that in a Raji lymphoma model, we could definitely image these tumors in vivo. We then did further studies on this in a melanoma tumor-bearing mouse model, and this is just a biodistribution study showing that this compound targets, and we get specific targeting in the tumor, spleen, and bone marrow, all of which are receptor or have high concentrations of alpha-4 beta-1. And if we add an excess of the ligand, we can block this uptake. So once we established that, yes, this molecule works, we can target alpha-4 beta-1, we then moved into the preclinical imaging of the hematopoietic progenitor cells. It's a mouse model of a human tumor bearing mouse model that this human tumor cell line, MDA-MB231, expresses luciferase. So we can actually monitor the metastatic process by just doing simple bioluminescent imaging. And so just what we did was we injected the tumor cells on day 1, and then we monitored them out to their growth of about 21 days. We looked at both bioluminescent imaging and PET. We did a pre-scan before the tumor cells, and then we monitored them on several days out. And so when you inject these cells intracardially, they actually will metastasize to the bone. And that was a model that we were familiar with, which is why we chose that. So what you see here then is the bioluminescent imaging. So the way you're probably familiar with bioluminescence, it's the cells are transfected with firefly luciferase. And when you implant the cells into the mouse, then you inject luciferin, and you have about a 15-minute window, and the luciferin will then just light up cells that express this gene. And so that will specifically then image these tumor cells. So we can just monitor when we get bone mets, which you can see here. So this is just bioluminescent or BLI imaging over time. And then we do the PET imaging, and we're focusing just really on what's going on here. The tumor cells themselves do not express the alpha-4 beta-1 or the VLA4. And this is just one animal where we do see, over time, we're seeing an increase in signal in this leg bone. And this, again, just to emphasize that the tumor cells do not express the integrin. And if you look then, if we take the bones out and do the histological validation, what we see is we see the alpha-4 beta-1 anti-alpha-4 antibody stain actually is in cells that are not tumor cells. So there are other types of cells which we are assuming are bone marrow-derived cells, hopefully the hematopoietic progenitor cells. So we did a series of mice, and we showed that the uptake increases over time in the bones, and we are able to we correlated this again with flow cytometry and showed that in animals where we actually get a high signal in the leg bones, we get increased numbers of hematopoietic progenitor cells. Here's an animal that has a higher SUV and we get a significant increase in the HPCs. Here's one where we don't see any bone mets, and we actually see a lower level of the HPCs that's closer to the background levels. And we showed that there is not a great correlation, but we're definitely seeing a correlation with standard uptake value, which is from the PET scan. And this is showing that this is a correlation between the standard uptake value from the PET scan with the bioluminescent imaging, showing that we typically end up seeing metastases by bioluminescence imaging. So we now, again, that wasn't the best compound. Our SUVs were pretty low on that. We've improved the compound just by changing the chelator. It turns out that we're actually hitting more receptors, although the affinity is unchanged. We're getting more receptors with this new ligand. Don't ask me why on that, but it seems to work. And we're getting higher uptake in our B16 model. So we've done some preliminary imaging. This is a different metastatic model where we inject, it's another breast cancer model, but it's a mouse mammary tumor cells. We inject them in the tail vein. They metastasize to the lung. And we're in these cells, this particular cell line 4T1 is VLA4 negative. And we're seeing increases again over time. We imaged before tumor and then 2 points after we injected the tumor cells. The tumor-naive mice do not change, and we see significant increases in the lung SUV. And this correlates as well with hematopoietic progenitor cells that are circulating in the blood. And there's a colony-forming assay that we did to correlate this. So, to summarize, what our goal is here is to image hematopoietic progenitor cells that are present in high concentrations in the pre-metastatic niche. I think we have data that suggests we're able to do this. I think we're hoping to try this more with our new compound that seems to hit more of these receptors. I think what I really wanted to stress that we have to use multiple modalities to really tackle this problem, and we have to validate everything with flow cytometry, cell sorting, and immunohistochemistry. Our preliminary data show that both of our compounds are taken up in sites of metastasis prior to and after the tumor cells are present. And we're just getting started now with our new imaging center to start looking at the 4T1 model and validate that in the lung met model. So finally, I just wanted to show you, this is our new imaging facility. It opened yesterday. It's in Hillman Cancer Center. So now under one roof at Hillman Cancer Center, there's preclinical MRI, ultrasound, 2 bioluminescent scanners, optical tomography, and PET-CT. So, and this is the new suite is the PET-CT, and this is the new, this one is bioluminescent with X-ray. So we have a nice big room back there. It's all behind the barrier, and we hired a person, Kim Ferraro. She's going to be running this facility. And we also have our animal holding rooms right there, so we have all our animals will be right next to where we're scanning. And finally, I just want to, this is the group that's been assembled in my lab over the last year at Pittsburgh. We just took that picture yesterday overlooking the Monongahela River there. And I'd like to acknowledge our funding sources and the generous startup we've gotten from both the University of Pittsburgh and UPMC. Just got some funding from DOE that I'm very excited about, and then Washington University, of course, where I worked for 20 years. Thank you for your attention.
Thank you very much. Really exciting, from the science to the funding, all good, right? Yeah. Thank you very much. Questions? Comments?
So in practice, how would this be used, fast forward?
Okay, yeah, so if this really did take off, I mean, I guess what we could envision is if, even with, well, what we would envision, I think, would be that if somebody is, they find out they have cancer, they get their primary tumor removed, and they may then go on to get one of these PET scans with the new agent to see if they have any pre-metastasis.
Right, and if that's the case, right, absolutely debris left over.
If that's the, yeah, if they do have pre-metastasis, there are actually agents out there that target some of these, there are drugs that are under development now that target alpha-4 beta-1 integrin and target the VEGF-R1 receptors. So they could be eligible, hopefully in the coming years there'll be drugs out there. I think that, it just allows those patients to really be monitored more closely.
And you could imagine once the biomarkers really are identified and it's proven that knowing of them will have therapeutic value, that then you could get into the era of thinking about simplifying regular monitoring, low-cost regular monitoring.
Well, and that's where I think in some cases optical may also come in. Although, some of the issues with optical are that, you can't go deep into the body. It'd be very tough to look for liver mets and lung mets. But this agent also has applications in lymphangiogenesis. And so you could look at, it turns out that the lymphatic endothelial cells that, how breast cancer will typically go to the lymph nodes first. So if you're in surgery, you could see if you have this lymphangiogenesis, which is another precursor to metastasis. So that's another avenue that we're going to be exploring. And that could be done, we could do a PET scan initially, but it could also be done optically.
Right. Other questions? Yes, back there. Yes, ma'am.
In response to your question, don't ask me why. Is it likely that the binding to the receptor has increased due to activation of any previously inactive receptors?
Oh, that's an interesting thought. Yeah, I've seen this with other receptor systems where we just change the molecule slightly, and we're not even touching the targeting molecule. We're just changing what's hanging off the end of it, and for some reason maybe the charge or something tends to make it more to bind better or to bind, not even better because it's not even binding with higher affinity, but to hit more receptors.
It is known that some receptors get activated in the presence of kinases. They add phosphate groups and become active. So you may want to look into kinases in that system. Okay, great, thank you.
Or to be less likely to be unbound by other.
Right.
Because you're looking downstream after it reached steady state.
Right, and we're just really looking at tumor cells at this point. So, yeah.
More questions or comments? Who agrees this has been a very interesting day and afternoon, particularly seeing how impressive these results are getting, moving towards clinical application, fundamentals, reinventing technologies from the scratch. Even some adjoint operators snuck in there, right? Kind of. Okay, I think all the speakers deserve a big round of applause. And remember, speakers, do not go wandering off or you will be lost and won't get dinner.